Good afternoon, everyone. I'm delighted to be here with Dr. Suzanne Tuff to speak about our collaboration on the Alberta Births Common Data, or ABCD study. Dr. Tuff is a professor in the Departments of Pediatrics and Community Health Sciences in the Cummings School of Medicine here at the University of Calgary. She's also the principal investigator of the All Our Families cohort, formerly called All Our Babies, a cohort established at roughly the same time as APrON. I am Nicole Letourneau, professor in the Faculty of Nursing and Departments of Pediatrics, Psychiatry, and Community Health Sciences in the Cummings School of Medicine here at the U of C. Our full biographical details are available on the conference website at apronstudy/conferences in the speaker booklet. Today, we're going to speak about collaboration between the APrON and All Our Families teams. We joined forces to collect the same data at 5, 8, and 12 years of child age. Our talk today will describe our study's independent and collective impacts and new directions. About 10 years ago, there were two large research initiatives underway studying families, and especially women and their babies over time. Dr. Suzanne Tuff led the one called AOF, and Dr. Bonnie Kaplan led the other called APrON. After Bonnie retired in 2013, I stepped in to lead the APrON team, and I've carried on to help realize APrON's vision. As both studies were wrapping up their first 3 or 4 years, Suzanne and I realized that the power of the information we were collecting could be multiplied many times over if we worked together. So we decided to join forces and move forward as Alberta Births Common Data. We were very pleased to receive $1.9 million from the Alberta Children's Hospital Foundation to undertake our ABCD research together. Not only did this enable us to collaborate on collecting the same data, the funding enabled our cohorts to continue. The importance of this support cannot be overstated. APrON and AOF are treasure troves, and in my opinion, and I think shared with Suzanne, an important Alberta legacy. You may be surprised to hear that APrON and All Our Families are two of the largest longitudinal pregnancy cohort studies in Canada. APrON is comprised of about 2,200 mothers and their children, and about 1,300 fathers. All Our Families is comprised of about 3,200 mothers and their children, for a total of about 5,400 Alberta families. Each study includes a diverse population and a large range of outcomes, including those related to birth and pregnancy, nutrition, mental health, community resources parents access, health services they use, parenting practices, social support available and relied upon, vaccinations, families' lifestyles and behaviors, their school and work, the quality of their relationships, and measures of their physical environments. The data has provided fantastic, rich information that has allowed researchers nationally and internationally to look at trends over time in mothers and developing children, and to examine family strengths and gaps in services in a typical Canadian urban city. We're convinced that this continued collection of longitudinal data helps researchers guide policymakers toward decisions that best meet the needs of families. Our unified goals are to create new information about child development to inform programs, policy, and practice, to add to All Our Families and APrON with common follow-up questionnaires for children at ages 5, 8, and 12 years of age and their parents, to increase research outputs through data sharing with highly qualified investigators with specialized expertise, and to advance community partnerships to enable efficient knowledge exchange.
To give you a sense of how our longitudinal research projects work, this graphic shows the timeline of data collection for the two projects led by Nicole and myself. In both projects, we recruited pregnant women who signed up, understanding that we wanted to study them and their babies over time, to see how biology and social environmental factors affect their health. We collected biologic samples like blood and urine, and asked them to fill in detailed questionnaires about everything from their diet and living conditions to stress. The first infant was recruited into All Our Families in 2008 and into APrON a year later. We are currently in the 8-year follow-up phase for over 5,000 families. We're pleased to report that we believe we're on the right track because we have a 70% retention rate overall. So why collect biologic samples and detailed information? And why over time as opposed to snapshots or cross-sectional data? Well, by understanding all of the factors that are part of a participant's lives, we are better able to establish what is truly cause and effect with respect to their health, So what comes first? Understanding the modifiable factors that influence development before the disorder is expressed provides insight into how we can intervene to prevent or reduce the severity of poor outcomes. Our data is forming the basis for a very broad range of studies, from how stress gets embedded in biology to how social support can offset the adverse effects of maternal depression on child and family well-being. Today, we're going to give you a couple of examples of how and why this information is so valuable. By way of illustration, our expertise and depth of information is highlighted on this slide here. You can see it ranges from experiences that happen in the prenatal period to those that happen once the baby is born and across early childhood. They range from physical factors to psychological factors to community resources and health and well-being in general. My first example of how we're using ABC data is quite current. Just as we were launching our project, personal computing devices were being introduced to the world. The iPhone made its debut in 2007, the iPad in 2010. So we began collecting data about kids' exposure to these devices and other forms of media. And we're glad we did, because in the years since, there has been lots of discussion and debate around the impact of those devices and social media on young people, including how excessive screen time impacts child development. Before I show you what we found, these are the current guidelines in place as recommended by the Canadian Pediatric Society. You can see that when children are younger than 2 years of age, no screens are best. From 2 to 5 years old, less than an hour a day, and at 6 years and up, parents should place consistent limits on time and types of media, ensuring that screen time does not interfere with important activities like sleep and exercise. Why should we care about screen time? Because it's been found already that children who exceed screen time guidelines are at increased risk for poor self-regulation. Things like being able to wait your turn or delay gratification. Children with poor self-regulation are more likely to have challenges in school and social activities, with evidence suggesting these challenges can persist into adulthood. Our study looked at the earliest exposures to screen time and the effect that has on child development. From this slide, you can see that this is the average amount of time children are spending on screens per day. This data suggests that 20% of infants are meeting the guidelines and 18% of children over the age of 6, and only 5% of those aged 2 to 5 are within the recommended limits. Consequently, more than 80% are exceeding the limits, which could threaten development and have a negative impact on the likelihood of good self-regulation. Our work suggests that addressing screen time could protect thousands of Canadian 5-year-olds from threats to development. We recommend families pay close attention to screen time, choose quality programming, and watch together. They may want to develop a family media plan. You may be interested to know that this study has received tremendous attention, including news stories in CNN and numerous views on the study website. I just want to comment here that in the presence of COVID, we're also aware that families are relying more on screens, and in this extenuating circumstance, we don't necessarily know that that's a threat to development. Screens are being used now for education and staying connected with families and in relationships, so stay tuned for more information on the impact of screen time and development. Example number 2 involves preterm birth. Did you know that Alberta has one of the highest rates of preterm birth in Canada? Almost 9% of babies in Alberta are born before 37 weeks gestation. These preterm infants often end up in the neonatal intensive care unit. They have a higher risk not only for significant health problems at birth, but also for longer-term health problems and issues with learning, behavior, and overall development. The earlier the delivery, the higher the risks. With limited ways to anticipate which moms will deliver preterm, we recruited 1,878 pregnant women to help us try to identify potential predictors. Of the 1,878 women, 102 delivered their babies preterm. Knowing this, we conducted thorough reviews of their medical charts and then worked with basic scientists to analyze their blood. We compared their results against mums who had delivered at term. We also identified genetic markers in the preterm mums, which they shared in common. The good news is that with this information, we've been able to develop a blood test that can help to identify who is more likely to deliver early. We received a lot of attention for this discovery, which is very nice, yet at the same time, we need to remind people that a blood test is only one piece of a much bigger puzzle. There are clinical variables such as who has delivered preterm before, who may have terminated pregnancies before, anemia, bleeding, urinary tract infections, and smoking, which on their own can predict about 41% of the risk for preterm delivery. The best prediction from genetic analysis through the blood test is about 62%. When we combine the clinical and genetic markers, our ability to accurately predict preterm delivery improves to almost 80%, a significant improvement that we hope will help women far beyond the borders of Alberta. Our next phase of work will be to determine if there are ways to lower the costs of this biologic analysis, to understand other clinical predictors, and to make this test more accessible.
This third example comes from APrON and speaks to even wider collaboration enabled by our work. We were able to be part of a study to predict epigenetic age in children. The term epigenetic refers to changes in genetic expression that are not affected by the underlying genome, or DNA. All of the human body's functions are dictated by proteins expressed by genes. And while your genotype does not change over your life, gene expression is changed from exposure to different experiences. One such epigenetic process is called DNA methylation. Epigenetic age, often referred to as DNA methylation age, has emerged as a highly accurate estimator of chronological age. Epigenetic age, if advanced beyond chronological age, may be viewed as a biomarker of stress exposure. However, current epigenetic clocks are not very accurate in the pediatric age range, perhaps because DNA methylation changes much faster and more frequently in children. If not for APrON's collection of blood and cheek cell samples from children at 3 months of age, we could not have contributed to this landmark study that created a pediatric epigenetic clock. Because the DNA was sourced from APrON children's cheek or buccal epithelial cells inside their mouths, the clock was called Pediatric Buccal Epithelial Clock, or PBE clock for short. Not very catchy, I know. The PBE clock was characterized in multiple cohorts, including APrON's, showcasing its accuracy in longitudinal data and associations with obstetric and child outcomes. Findings show that the PBE clock accurately predicted chronological age of children across a range of ages, much better than the adult epigenetic clock. Infants' gestational age was associated with the PBE age when children were 3 and 9 months of age. And the PBE age in children with a neurodevelopmental disorder, autism spectrum disorder, showed a higher PBE age than those considered to be typically developing. Because we produced so many studies in APrON showing impacts of maternal depression on child health and development, and these example papers are literally just a few, a big part of my research program has been to use that knowledge to work with Calgary and international partners to help mothers affected by depression so that children's development is protected and enhanced. These APrON studies on impacts of maternal postpartum depression on children led to the CIHR-funded VidKids study. VidKids is short for Video Interaction Guidance for Mothers with Postpartum Depression and Their Infants. It's a trial of an intervention for mothers affected by postpartum depression conducted in partnership with Alberta Health Services and the University of Washington's Parent-Child Relationships programs at the Barnard Center. In our VidKids study, we offer support to mothers so that they can interact with their infants in ways that will protect their development. As you can see from the left column, our goals are to reduce mothers' symptoms of depression, improve mothers' sensitivity to their infants' cues, and reduce both mothers' and infant stress levels, assessed via the stress hormone cortisol. The intervention is comprised of 3 sessions and 3 home visits provided by registered nurses. And by the way, now we are converting our program to online delivery, thanks to a CIHR COVID-19 grant. This will make the program more available. We hypothesize that improving maternal-infant interaction quality will reduce infant stress and maternal depression, thereby improving infant developmental outcomes. Our published pilot data show that the program significantly improves parent-child interaction, especially maternal sensitivity to cues and maternal activities that promote cognitive growth in the infant, as well as reduce infant cortisol levels. We approach significance in impacts on maternal depressive symptoms, but it should be noted that the effect sizes for all 3 variables were moderate to very large, that is 0.42 to 1.43. We think that with a larger sample size, we will be able to detect impacts of intervention on the mother's depressive symptoms too. So, in our follow-up CIHR-funded trial, we've already recruited 120 families and are targeting about 100 to 130 more. We will also, we hope, examine the children's brain development via MRI in partnership with APrON team member Catherine LaBelle and are seeking funding for that now.
We've worked together to establish an excellent COVID-19 collaboration to understand the impacts of the pandemic on children and family in terms of risk and resilience. We've retrieved data from over 1,800 families across the cohorts, and we've collected this data between May and July of 2020 with the intention of collecting some additional data as the pandemic unfolds. We're independently collecting data on children's experiences directly from the children for the first time, with questionnaires tailored to the ages of our sample. Although we are aware that families are doing their best to cope during this unprecedented pandemic, and many are finding avenues of resilience and using their imagination to keep routine and structure in their daily lives, we also know that many families are challenged. Our information revealed that 40% of urban Albertan families report a reduction in income through job loss or hourly reductions. Up to 30% of mothers report elevated stress, anxiety, and depression, twice the rates we would typically anticipate. Over 40% of our families say that their relationship includes partnership tension, again, twice what we would anticipate typically. Almost 30% of mothers are concerned about their child's behavior and mood, although the vast majority anticipate that their children will be successful at school. We notice that our findings mirror the results from the Ontario Parent Survey.
We are hoping that these examples give you a sense of the valuable work possible with such precious cohorts. There are very few longitudinal studies like ours in the world. Some of our newest faculty at the University of Calgary have joined our teams in order to work directly with us. This amazing diversity of research inquiry would be unimaginable without the infrastructure support to maintain the highest standards of data management and ongoing data collection that we have been able to achieve with the support of the Alberta Children's Hospital Foundation. And going back to our project goals, I hope you can see that we are indeed conducting quality research about child development that's contributing to advancements in practice, policy, and research. Our data is so rich and holds so much potential that we have more than 108 researchers from across Canada and internationally using it in more than 140 studies. We have produced nearly 200 papers combined to date. Just in this last year alone, our researchers have been awarded more than $2 million in grants to continue their work. Over the years, researchers using the ABC data from both other families and APrON have received well over $23 million over and above the original contributions from the Alberta Innovates. And finally, going back to ABCD's third goal, to influence public policy, what we have been given is the opportunity to gather contemporary data that is integral to informing and shaping program planning and policy. We are being invited to sit at the table with government and community groups as they tackle where to make healthcare and programmatic investments, how to develop clinical practice guidelines, and how to respond to issues affecting families in our communities. Evidence is so crucial in developing social strategies. We are now gathering that evidence and have the information to help identify children and families currently at risk of developmental issues, reduce and prevent risks in the future, sit at policy and strategic planning tables to ensure evidence is available to inform decisions, and to develop interventions that can strengthen children and family success in the long term. Follow-up work in APrON particularly addresses the intervention component, with my own projects designed to help mothers with depression or who are exposed to chronic stress serving as the best examples. In our continued work together, we've recently embarked on a new proposal for a project with a huge transformative aim. Together, we intend to transform mental health care for children and adolescents in Canada and internationally by developing, testing, scaling, and spreading the implementation of a screening and recommendation tool to prevent children's mental health problems. We expect clinicians will use the tool to identify children and adolescents at risk and make decisions about or recommend access to services to prevent mental health problems, thereby reducing suffering and societal burden. We have applied for about a $20 million grant to do this work with the New Frontiers Research Fund, with partnerships between us at APrON, all our families, and an Australian cohort called the Bar1 Infant Study, or BIS cohort. And how do we plan to do this? By using all of our amazing data over time to develop a better model for predicting risk for mental health problems in children and youth. And this information should ultimately enable targeted services and supports to prevent children's mental health problems in the first place. For this proposal, we want to capitalize on our genetic and epigenetic data in APrON, All Our Families, and BIS, noted in the slide in purple and green, as well as extensive data on health, noted in the slide in orange, including nutrition, neurotoxicants, and family health factors, and more, as well as data on social factors in red on the slide, including parental distress like depression, social determinants of health, and parent-child relationship qualities. With data on these factors extending from prenatal life to when children are 12 years of age, and using sophisticated machine learning techniques, thanks to our newest team members, Niels Folkert and others, we project we will be able to develop a truly comprehensive and breakthrough approach to predicting risk for mental health problems in children and enabling better prevention. This is an important undertaking given the dramatic rise in mental health disorders in children and youth that was apparent before the pandemic and that have become even more prominent since the pandemic. Our proposed work is essentially a grand challenge that puts together our past successes with social genetics and epigenetics, successful health and social data collection over time, quality data management practices, lab science capacity, and other successes with promising new directions, especially in using machine learning to understand children's health and development, and so inform policy and programs to address the growing chorus of concerns about children's and adolescents' mental health problems. The APrON and All Our Families data and combined ABC data has provided fantastic, rich information that has allowed researchers nationally and internationally to examine predictors of children's health and development, examine trends over time in mothers and developing children, examine family strengths, and finally examine gaps in services. We have featured just a few of our APrON and All Our Families colleagues here who have conducted this impressive research. We are convinced that this continued collection of longitudinal data will help researchers guide policymakers toward decisions that best meet the needs of families. We look forward to many more years with our APrON and All Our Families. Thank you so much for listening. We're happy to answer any questions now.